Hello, this is the process of meiosis. The process begins with interphase, where the DNA strands grow and duplicate into sister chromatids, as can be seen here. To the left, the blue and gold sparkly pipe cleaners represent the DNA for sperm cells. On the right, the blue and pink fuzzy pipe cleaners are used to model DNA for egg cells. Homologous chromosomes can be seen with the same different colored, same-sized pipe cleaners. The first phase of meiosis is prophase 1, where the DNA joins together using centromeres to create chromosomes. These chromosomes then perform the process of crossing over, where the chromosomes will exchange DNA, creating genetic variation and genetically non-identical daughter cells. Next comes metaphase 1, where a pair of homologous maternal and paternal chromosomes will come together through their centromeres and join, forming a tetrad. This is also when independent assortment occurs, where the chromosomes align and pair up with other chromosomes in varying orders. This will determine DNA and genetic variation the resulting cell will end up with. Next is anaphase 1, where the tetrads are pulled apart by spindle fibers and are now separated on opposite ends of the sperm and egg cell. After anaphase 1, telophase 1 is where the nuclear membrane reforms and the divided chromosomes are now surrounded by the nucleus. Each original parent cell will result in two daughter cells in the final phase of meiosis 1, cytokinesis 1. Now that we have two daughter cells for each, both the egg and the sperm, meiosis 2 can occur. It first begins with prophase 1, which is much less eventful than prophase 2, since the newly formed cells simply prepare for division in the next phases. The nuclear membrane will break down and allow for full separation of the DNA. Metaphase 2 is similar to metaphase 1, but instead of aligning the tetrads, which are made up of two chromosomes, we are aligning individual chromosomes in the cells. Next, anaphase 1 is also very similar to the process of anaphase 1, but instead of separating the chromosomes in their tetrad form, we are separating the individual chromatids within each chromosome using spindle fibers. Each separated chromatid becomes an individual chromosome. Now, in all of the cells, two maternal and two paternal, the chromosomes are on opposite ends of the cell. The nuclear envelope will begin to reform in the next phase, telophase 2. The newly separated DNA groups together in each of the new nuclei. Cytokinesis 2 is the final stage of meiosis. All cells pinch down the middle, separating the cytoplasm and ultimately dividing each cell into two daughter cells. Because this occurs to two cells in both male and female organisms, it results in 4 genetically different cells each. After meiosis 2 has occurred and the sex cells, or gametes, are produced, fertilization can begin, where a sperm cell from the male will unite with and fertilize an egg cell in a female organism. This union forms what is known as a zygote, which will divide and eventually become an embryo, which will result in a brand new organism. As you can see, meiosis plays a crucial role in sexual reproduction and the multiplying of organisms and populations. Thank you.